Today we're going to be talking about the cell cycle. Remember, you guys are going to generate some nice detailed notes, a 3-sentence summary, and you're going to have some questions of anything you don't understand. What is the cell cycle is where we're going to begin. And so that we have to kind of jog our memories all the way back to the beginning of the year when we talked about what consists of a living thing. Living things, if you remember, are all made up of cells. And we know that these cells must come from pre-existing cells. In order for us to get pre-existing cells, we have to ask ourselves 2 questions. Why do organisms need new cells, and how do organisms need new cells? So, first question is going to be, why do organisms need cell division? And first is an obvious reason, is for reproduction. We have single-cell organisms and complex organisms. Single-cell organisms are generally going to go through something called asexual reproduction, where more complex organisms like us are going to go through sexual reproduction, where you have 2 individuals creating a new, whereas in asexual is 1 organism creating an exact copy. Second reason we want to have cell division is because of growth. You are not the same size, or do not have the same number of cells in your body that you do when you were born. So you've increased your cell number, you've grown. The third reason we have new cells is because of regeneration. You guys have fallen, cut, skin your cells, and you need to repair and replace them. Old cells wear out, you have to have new ones. There are organisms that can actually regenerate new body parts as well. So we have regeneration. So we're going to start with looking at how do the simplest organisms reproduce. And that would be our prokaryotic cells. And prokaryotic cells reproduce by something called binary fission. And this is a form of asexual reproduction where you basically have a single parent creating an exact copy. This is mainly done by bacteria and simple single-cell organisms like amoebas. Now, binary fission, by definition, is 1 parent creating an identical offspring. Well, let's look at how does that occur. So we have here in this picture a picture of a prokaryotic cell with its single strand of DNA all coiled up inside. What happens is this single strand of DNA is going to copy itself and replicate itself. It's going to take up a lot of space, so the cell is going to elongate itself out as it does. Then they're going to separate and pinch each other off so you have a completion of the 2 daughter cells. We use the term daughter cells because you're creating a new generation, not to talk about gender. It's just a term we use. The next, let's talk eukaryotic cells. Eukaryotic cells, if you remember, they're what we're made up of, much more complex and generally have way more than 1 single strand of DNA. Matter of fact, we have 46 strands of DNA. So there's a lot going on that we have to copy. So in our cells, let's, before we begin, talk a little bit about DNA. If you remember, DNA is a double helix made up of nucleotides. And over here we have a nice picture of our double helix. And when we talk about it, we're going to talk about the DNA coming in different forms. So when we see DNA loosely packed with the histones, these are the little small balls here, they're proteins that hold the long strings together. We're going to call it chromatids. And then when it's all tightly coiled up, we're going to call it chromatids. Then when we have 2 of them together, 2 chromatids together, held tightly together by a centomere, we're going to call it a chromosome. And this little piece here, the indentation, is what our centromere is. So we have many, many different terms for this material, but it's all in fact DNA. It's kind of like you have a crumpled up piece of paper. It's still a piece of paper, it's just a different form. So you will be responsible to make sure that you can identify when something is a chromatid, when is it a chromatid, and when is it a chromosome, and where is the centomere located. So take a good look at those pictures, draw them out in your notes if you have to, label them so that you'll know them, because you're going to really need to know that. So let's get on to the cell cycle. So the cell cycle is basically divided up into 3 major phases. And just like the life cycle, it is an ordered sequence that has a beginning and an end. And so the first part is the beginning, which is interphase, which the cell spends approximately 90% of its time. And it has divided up into 3 subsections, G1, the S, and the G2. Second phase is the mitosis phase. It's much shorter, but this is where we have nuclear division. that's where the chromosomes are going to divide and separate themselves out from each other. Then the last stage is cytokinesis, and this is where we have the cytoplasm is going to pull apart, and you're going to end up with your two nice daughter cells. So let's take a minute and take a closer look at interphase and its subphases. So in the G1 phase here, this is when the cell is going to grow. It's normal activity. And then in the next part of it, it's going to be called the S phase, or the synthesis phase, and this is where DNA makes an exact copy of itself. So all those strands of chromosomes, we start out with 46, we're going to have 92. So you've got a doubling of all of your DNA. And the third phase is the G2 phase, and this is another growth phase where you're really basically preparing for mitosis and cellular nuclear division. So that brings us to mitosis. Let's take a peek. So in mitosis, we talk about it being, by definition, nuclear division. So again, it's going to be something that's divided up into more than one phase. We'll get to that in the next video. But basically, in your cell, you have 46 chromosomes. During the S phase, it's going to jump to 92 because you've copied them all. During nuclear division, you're going to take those copies and make sure that they equally divide into the new, potentially new cells. So you're going to separate them out into the 46 again. Then in cytokinesis, you're going to have the splitting of the cytoplasm. Now, when we talk about cytokinesis, we have to talk about, again, we're in eukaryotic cells, and remember, we have two types of eukaryotic cells. We have plant cells and animal cells. Because they're slightly different, we have to explain how cytokinesis is slightly different. And in animal cells, you're going to have these group of proteins that are going to adhere together and kind of pinching, kind of like a drawstring, and it's going to be called a cleavage furrow. This will then pinch off between the two cells, creating our nice new daughter cells. Plant cells, however, remember, they have a cell wall, so they have to do it a little bit differently. They have these little vesicles that are going to line up and connect to the plasma membrane in the cell wall on the outside, creating a new cell plate, which will eventually lead to the cell wall, and so that will pinch off and divide between your two daughter cells. Now, that's in general if everything goes okay in all cells. Fortunately, however, not everything goes okay, and sometimes things can get a little bit out of control. And that's when you really have cancer cells. So what happens is in your cells you have these checkpoints, and these checkpoints, the first one occurs after the G1. It's checking to see that everything's going okay, and if it is, then it goes ahead and goes into the S phase and copies your DNA. If that goes okay, then it's going to move into the G2 phase, and there's another checkpoint at the end here to make sure that the DNA has been copied correctly. If it has, it goes on to nuclear division, which is your mitosis. At the end of mitosis, if everything is divided out equally and you get your two cells, then cytokinesis can occur and you get your two new cells. Unfortunately, however, sometimes these checkpoints get shut down and the cell will divide uncontrollably. And that's usually what you have with cancer. And this cell would divide and divide and divide. And the checkpoints are completely pretty much shut off. And so it continues to divide until you have numerous amounts of cells. And so, for example, if you look down here, you have a cancer cell that divides and grows into a tumor cell. It keeps going until it gets larger and larger, and then eventually it's going to get into your bloodstream and your lymph system, and then it moves to another area of your body. And that can create all sorts of problems. Most cells, in general, only divide approximately 50 times, and then they stop, and they are destroyed. But here, cancer cells continue to reproduce, and this gets to be a huge problem and can eventually, unfortunately, lead to death. And that basically, in a nutshell, is the cell cycle. So let's just talk a little bit about here. And we have the major reasons we have cell division is for replacement, growth, and reproduction. And we can have reproduction in the form of asexual reproduction or sexual reproduction. So make sure you double check all your notes, go through this video if you have to. You want to make sure that you have in all of your notes, why do we have cell division? How do single cell organisms reproduce in the form of prokaryotic cells with asexual reproduction or binary fission? How do eukaryotic cells reproduce with sexual reproduction? And the DNA terms and all of their diagrams. Good idea of the cell cycle, all the stages and the end results. And lastly, you have a good understanding of what a cancer cell is. Thanks, and hopefully you come up with a good question tomorrow.